In previous lectures, we've addressed the processes and components of inflammation and immunity. Now we're going to think about alterations in immunity and inflammation. We're going to be taking generally that positive protective response and seeing how it may cause threats to homeostasis. So we're going to see how this applies. We're going to look at the mechanisms of immune system function and dysfunction, and we'll look at it in three exemplars or examples. We'll look at allergic response, which is looking at those negative effects of a hypersensitivity response or hypersensitivity, especially to environmental but other allergens. We'll take a look at transplant rejection, organ transplant rejection. This is an example of alloimmunity, where one individual, the recipient's immune system, reacts against another individual, the donor antigens. We'll also then take a look at an example of autoimmunity, systemic lupus erythematosus, in which the immune system in an autoimmune disorder, lupus is the example we're going to use, but in that example, the immune system isn't able to tolerate itself, and as a result, in an autoimmune disease, the individual develops autoantibodies and or autoreacting T cells. And so this idea of autoimmune diseases that we're going to talk about, it's very important because there are numerous autoimmune-based diseases, and we'll talk about them in many of the body systems as we move through the semester. So let's start by talking about an allergic response, which is an IgE-mediated hypersensitivity. So we know that this is part of our adaptive immunity response, and it is antigen specific. Remember that IgE is a class of one of our immunoglobulins. What happens in an allergic response, this IgE mediated, is the IgE actually binds to mast cell receptors. As a result, then the mast cell degranulates and releases or causes response, and the response is the production and the release of basophils and eosinophils. Now remember, when we talked earlier about eosinophils, we identified that eosinophils were part of our leukocyte cells that responded in terms of both parasitic infections, but also importantly in allergy. So as a result of the degranulation of the mast cells, as you know, this stimulates an acute inflammatory response in which histamine will dominate. In most acute inflammatory responses, especially IgE-mediated, H1 receptor response is greater than the H2 receptor responses. As a result, there's smooth muscle contraction, and the bronchus will constrict. There's increased vascular permeability leading to edema, as well as dilation, vascular dilation. This then leads to the redness that you'll see, cutaneous redness that you'll see within an allergic response, or hypotension if this is a severe allergic response. If this allergic response actually works well, and the H2 receptors will work to decrease the histamine from the mast cells so that you can start turning that off a little bit, because we know that ongoing stimulation of the H1 receptors with ongoing smooth muscle contraction and increased vascular permeability and dilation can be a threat to homeostasis. So when you're thinking about the allergic response, the IgE-mediated response, think about mast cells and histamine. So let's take a look at two examples. One would be an environmental allergy, the other would be a bee sting, because you'll see these in your practice. In an environmental allergy, it is possible that in some cases the individual is actually genetically predisposed, and this is called an atopic allergy. And so this is where you might see that you'll see allergic responses tracking through generations. What happens in an environmental allergy is you have an increased number of IgE globulins and receptors on those mast cells. As that histamine is released by the mast cells, at the point of antigen where it enters, you start seeing that response. So this is a key to most environmental allergens. The histamine is released by the mast cells at the point of where the antigen or the allergen actually entered the body. So it could be inhaled, it could be ingested, it could be by contact. Histamine, as you well know, starts that inflammatory process, dilation, and in both the GI tract as well as the respiratory tract, hypersecretion of mucus. So that's what's causing the IgE-mediated response. So when you think about that, think then too, what would the role of desensitization in an IgE hypersensitivity be? So why do allergy shots work on environmental allergies? Well, the idea behind that desensitization is for, as you give ever-increasing amounts of the allergen, the organism, the individual, generates IgG. That IgG that's generated then blocks and competes with the antigen. It prevents the allergen from binding with IgE on the mast cells. That's why allergy shots work. Another great example is a bee sting. We know that with a bee sting, you have both a local and a systemic reaction to the antigen. This is a little different. Remember, with an environmental allergy, generally you're going to see histamine released at the point of the antigen entry. However, with a bee sting, you have a larger systemic reaction. And this happens because with a bee sting, that stinger of the bee actually releases enzymes. That's why if you have a bee sting, you don't pull the stinger out, because when you pull the stinger out, it actually releases more enzymes. Now, the risk of any allergy, but certainly with a bee sting, is anaphylaxis. Anaphylaxis is an exaggerated, sustained response that is both local and systemic to the allergen, the antigen. Generally, you will see in an anaphylaxis, you have a very strong respiratory and vascular response, but you may also have a very strong GI response as well. So then, again, let's think about this a little bit, and sometimes it helps to put it in terms of therapy. Why does epinephrine reverse these local and systemic reactions? Because it reverses the histamine effects. Antihistamines in anaphylaxis do not work. They're not useful because the H1 receptors are all full of the antigen itself. So epi reverses the histamine effects, and antihistamine isn't useful because an antihistamine can't shove anything off those H1 receptors that's causing the problem. Let's look at an autoimmune example. Systemic lupus erythematosus is a good example of an immune complex-mediated hypersensitivity. And this is terminology that your text uses, so I wanted to be consistent. What happens in a general, and let's talk first about general concepts of an immune complex-mediated reaction. What happens in immune complex disorders is that the antigen-antibody complex is actually formed in the circulation. So you've got complexes being formed in the circulation. Those complexes then present to a target tissue, and that then activates the complement. The end result of this complex presenting to and then activating the complement is that the complexes overwhelm the ability of the neutrophils to phagocytize, and as a result then lysosomes are released into the tissue. That then causes tissue injury and damage. The other thing that's very important in these immune complex disorders, and remember this is primarily autoimmune, is that it tends not to be organ specific. Some are, but most are not organ specific. This complement activation leads to damage and harm because it's deposited in numerous tissues. The other thing that's very interesting about autoimmune or immune complex diseases is that they have a remission exacerbation characteristic. So it's almost as though the immune system has the ability over time in the exacerbation, or if we provide therapy, we can start to tamp down that complement activation, and we can start to diminish the formation of antigen-antibody complexes within the circulation. Once we do that, then the disease enters a remission phase or has a much, much lower period of activity. However, something then triggers another exacerbation, and so you have frequent exacerbations and remissions. In an autoimmune disease, in many, many cases, not always, but in many, many cases, because there is so much potential end organ damage, each exacerbation causes a little more damage until the organs have major disease. Let's take a specific look, though, at lupus, or systemic lupus erythematosus. Lupus is an autoimmune disease in which circulating immune complexes with anti-DNA deposits in a variety of tissues. So you have these circulating antigen-antibody complexes that have formed in the circulation, and they formed because the individual has for some idea decided that their own DNA isn't their own. So it's autoimmune because the DNA is the individual's own DNA. It's not coming from the outside like you had with that allergic response. It came from the outside. And this anti-nuclear antibody is very, very important. Lupus affects a variety of tissues, therefore there's that systemic part. It affects kidneys, heart, and vessels, causing a vasculitis. It can cause an arthritis, which is connective tissue, and it can actually cause a cerebritis and difficulty with the brain. So that means that these complexes have the ability then to breach our protective blood-brain barrier. Like many autoimmune diseases, lupus is progressive. It usually leads to slow end organ damage, but since tissue damage is cumulative, the tissues begin to lose their functional ability over time. So control of the immune complexes and control of the reaction becomes very, very important. If you take that then to a therapeutic range, just to understand how we manage this, again, I'm not going to test you on management, but I think it helps us understand that in order to decrease the exacerbations and the damage of this circulating immune complex disease, we have to then modulate the immune system. We have to suppress those T and B cells, because when we do that, we suppress complement that is the downstream effect of those antigen-antibody responses. You're going to be spending a lot more time with this disorder when you do your case study. The third example is an example of alloimmunity, which is transplant rejection. And this is more of a cell-mediated hypersensitivity. When we talked about the immune system, we talked about the major histocompatibility molecules, the MHC, and these molecules are present on the surface of all cells except those red cells. These molecules are genetically coded and they're incredibly diverse. There's a huge number of different alleles, and remember, you would inherit one allele from each parent. Their function is to present the antigens to the immune system for response. The immune system then recognizes that potential danger. Primarily with a rejection episode, with the MHC system involves lymphocytes, especially cytotoxic T lymphocytes. But you also have that role of the T helper cells as well. That's why when we look at a transplant rejection, this is an example of that cell-mediated hypersensitivity, the book that McCants and Huther discuss. Because it is cell-mediated, and we know that our T cells are heavily involved, you know that it is characterized by direct cellular destruction. Now, this mechanism then becomes pathologic in a transplant rejection. Every individual has a different set of MHC surface antigens. Remember, there's a huge diversity, a lot of variety, and it's genetically coded. The antigen in a transplant rejection is human lymphocyte, are the human lymphocyte antigens. This is a pretty big family, but we usually measure about 6. The one that we're the most familiar with is HLA, human lymphocyte antigen DR, and it tends to be pretty important. We also know that although the MHC molecules are very diverse, over 700, there's less diversity at the level of the antigens that are recognized. So what happens in a transplant rejection is that the recipient either has or develops antibodies to the donor antigen, and the speed at which they develop antibodies will vary. If the recipient has preformed antibodies to the donor antigen, this is what they call the hyperacute or immediate rejection, because remember that idea of antibodies have memory. And the recipient, in a transplant rejection, the recipient could have developed antibodies to the donor antigen through blood transfusions, as an example, or in another way. They wouldn't develop antibodies to a family member as likely, because they would tend to have the same antigens. They would share antigens because of that MHC, that HLA system. In a transplant rejection, the recipient develops cytotoxic T cells and T helper cells in response to the donor cells. So again, typical cell-mediated T cell response. The T helper cells release cytokines, which then activate the macrophages that are infiltrative. Those macrophages infiltrate, and the killer cells then attack the vascular structures, the endothelial cells of the donor organ. And that's how transplant rejection occurs. So what do we do about that? Well, we suppress that T cell response. Chronic rejection is due to either an ongoing inflammatory damage of the endothelial cells or activation of complement. So acute rejection is different than chronic rejection. Chronic rejection occurs over time and is very difficult to reverse. Let's end our discussion of this section of this dysfunction session and think about some of the physiologic effects on the immune system. And let's think about things that could actually decrease the ability to respond in the immune system. One of the things that we know that has negative physiologic effects on the immune system is stress. Stress actually decreases adhesion molecules as well as neutrophil chemotaxis. So as a result then, when the organism is under stress, remember that opsonization, that recognition and adhesion is very, very important. And of course, the ability of the neutrophils to get to the site of cell injury and get to the site of the immune response that's needed is slowed. Nutrition plays a key role in our ability to respond, our immune system to respond. We need nutrition for protein synthesis. Remember, antibodies are proteins. We need trace minerals for enzyme production, like zinc. We need vitamins for T and B cell function. So our nutrition supports our immune system function. If your patient has a malignancy, they are most likely producing cytokines and vascular endothelial growth factors. These cytokines and vascular endothelial growth factors can decrease the ability of the immune system to respond appropriately, because those cytokines and growth factors are coming from the malignant cells. And last but not least, certainly we induce negative effects on the immune system through our therapies. Our treatments, we've talked about cancer therapies before, the idea in a cancer therapy, but some of our other therapies, we actually target both healthy as well as abnormal cells. And when we do that, we're killing off our good stuff as well as the fast-growing cancer cells. We know that when we therapeutically alter our immune system to suppress our immune system, like in a transplant organ therapy, our patients are more likely to develop opportunistic infections. Remember, T cell dysfunction is manifested in a pattern of opportunistic infections. And then steroid therapy also decreases in a variety of steps our white cell function and our immune response. So as we bring this piece to a close then, we know that antigens can be exogenous, like a pollen, bee sting, another person's organ, or an antigen can be myself. It can be endogenous, where I recognize myself as not me. We know that therapeutic interventions can modulate or block these responses. And last but not least, remember that inflammation and the adaptive immune responses are intertwined, and they act together to protect us from a huge variety of assaults by antigens every day. I hope this has been helpful in pulling those concepts of inflammation and some of the concepts of the immune system together. If you have any questions, please be sure to let me know. Thank you.